So we continue our discussion on whole genome sequencing and the quality or whole genome sequencing towards the genome assembly. And what we're looking at is recognizing and trying to measure the quality of that genome assembly that we have done. So there is this BUSCO method or tool. It is widely used to assess the quality of genome assemblies. It stands for benchmarking universal single copy orthologs. What you find here is capturing some statistics from their database. In other words, this method, which is rooted in the concept of universal single copy orthologs. So when we talk about orthologs, we know that we are referring to genes, common genes from different species, these genes that are highly conserved across a wide range of species. And not only this, so they are trying to document here the genes. Okay, we have genome assembly. We're trying to measure the quality of that genome assembly. So what do we want to do? The idea is, let's zoom in into what we already documented from our knowledge across species, especially about orthologs. We know that the orthologs should be common, should be there to a certain extent. So if your own genome assembly is able to recover those orthologs, so instead, this is one way. It's one way to measure the quality. If I'm able to recover these orthologs across that we know from different species, of course, we need a database. Of course, this tool has its own database. Imagine we are working now with vertebrate, arthropod, fungi, these different groups of species, and then we focus on these orthologs. Not only this, there are single copies of some orthologs. So we don't want to end up just, with the very common orthologs. Another way, I have this genome that I've constructed. My genome is a good quality as long as able to recover some of these orthologs. Now, of course, you may say, well, it's not necessary that I have many of those orthologs, but again, we have a group of species already documented. You can look based on the group. Of course, this requires some design and special care when we look into this way of measuring the quality. So I hope you are able to get this idea, specifically knowing that BUSCO has its own databases that are lineage specific. So they have different sets of orthologs for different taxonomic groups, and then you would choose the relevant maybe taxonomic group if you're working with bacteria, fungi, or mammals, and so on and so forth. Now, the sampling space defines which organisms' genomes were used to determine the universal genes for a particular lineage. I think it's reasonable. We assume that there are universal genes. The question is, your genome assembly outcome, did it recover any of those? And not any, meaning, we're going to look into just any copy of those, okay? Specifically the ones that are having single copies across their genomes. When we talk about high universality of high universality of orthologs groups, the idea behind this is as follows. If an assembly or transcriptome is of high quality, then it should contain these highly conserved genes. If many of these genes are missing, this might indicate that there are gaps or errors in the assembly. And again, BUSCO focuses on single copy orthologs. These are just the statistics about across the species they have. So single copy genes is also what they focus on. These are the genes, again, across many species, typically exist as only one copy in the genome. So this is an emphasis, again, it's a way to, in essence, find and evaluate the genome, transcriptome, or gene set completeness by searching in this database and looking for the universal single copy orthologs. For variant calling workflow, because when I recognize the different variants, we can, of course, we are dealing with raw data first, doing the alignment and so on and so forth. And once we are done with these stages, right, we discussed before these steps, there are different ways or strategies into how you can look into the variant calling specifically. So there's joint variant calling, which refers to the process of calling variants on multiple samples simultaneously. We're talking here about, for example, father and the mother BAM files and so on and so forth. This is specifically relevant towards analyzing samples in cohort studies or family studies where we have multiple individuals or multiple individuals are sequenced. And there is also somatic variant calling. This is, again, as mentioned earlier, is relevant to cancer genomics. So the genome of cancer cells often accumulates some mutations that are not present in the normal cell, which is going to get us to some question. Is there a way to capture the genome of a single cell? There we're going to look into the single cell genome sequencing. That is very interesting area and very interesting development of technology. Something we will cover up as well in the coming lecture, which is single cell sequencing. Again, the cancer cells are having some mutations as compared to the normal cells, and these mutations are termed somatic mutations. So in terms of somatic variant calling, it's a process to identify these mutations by what? Comparing the sequence data from the tumor cells to that of the normal ones. Now, one of the terms here, which is an interesting term, is orthogonal validation. Now, this is simply involves confirming the results obtained from one method by using an entirely different. That is why it's called orthogonal. It's a totally different method. For example, if a particular variant is defined using next generation sequencing, it might be validated using Sanger sequencing, PCR, maybe whatever other method, and so on and so forth. So again, this brings us in a natural development of the discussion to single cell whole genome sequencing. It is focused on the idea of looking into the genomic information or content of individual cells. Now, compared to what was done before as whole genome sequencing, that is based on traditional bulk sequencing, where you take a pooled DNA from many cells. That is how DNA is extracted usually. So what about actually zooming into one specific cell at a time and then dealing with results looking at specifically the genome from this specific cell? So single cell sequencing isolates and sequences DNA from the individual cells, revealing cell-to-cell genomic variations. So this would indeed have lead to a specific recipes, specific methods. In general, again, the concepts of whole genome sequencing would certainly share many of the ideas with such different way like single cell. Of course, when you talk about library preparation, there will be some differences, right? Cell isolation, for example, this is one first step where some different fluorescence techniques are used, like FACS fluorescence activated cell sourcing. And then there is the whole genome amplification. Remember the cloning ideas there, because when we are working with one cell, what is the content of what would you imagine the content of DNA there? It's 6 picogram of DNA. So it is insufficient for standard sequencing procedure. That is why there is a need for some amplification strategy. So there would be some unique parts that will make up the process. But again, by the end of the day, there are going to be library preparation, sequencing, variant calling, analysis, and so on and so forth, validation. Okay, so in terms of also library preparation, as we say, we see here, there's the direct library preparation, which is a method to help simplify preparing the DNA for sequencing. In the context of this single cell genomics, it can minimize this method DNA loss. It includes some steps like using I5 and I7 adapters, which is part of the Illumina's dual indexing system, and that is used for multiplexing multiple samples in a single sequencing run. So also there is this concept of single cell combinatorial indexing, and this is a technique to tag molecules from individual cells with unique combinations of barcodes. Again, we are dealing with multiple cells. There is, of course, some level of complexity in preparing that library, but this is what's going to allow for the simultaneous sequencing of many cells, while still being able to identify the cell of origin for that specific or for each read. Symmetrical strand, single cell whole genome sequencing refers to a specific approach in the library preparation where both strands of the DNA molecules are used symmetrically, okay, where both strands are used symmetrically. And towards finally, that's gonna be the analysis. So there are unique steps into the library preparation where, you know, the biotechnician would be caring about. There's no question that single cell whole genome sequencing has lots of significance. For example, heterogeneity analysis, we'll see some examples as in the coming slides. So one of the primary advantages of single cell whole genome sequencing is its ability to uncover genomic heterogeneity. So for example, tumors are heterogeneous with different cells potentially maybe having different sets of mutations. This can affect the treatment and understanding the disease and the disease progression itself. So having this technology where we can zoom into the cell level, that is amazing. And trying to understand that, you know, identifying rare cells, looking into different things, and many applications are there for single cell whole genome sequencing, trying to derive the whole genome towards more studies. And in terms of applications of single cell sequencing, okay, we can use it to resolve complex genomic regions. As you see, we are now aligning these reads, and we're trying to, you know, based on this whole genome sequencing at the single cell level, we're trying to resolve complex genomic regions. For example, repetitive elements. Those might be difficult to assemble using the bulk sequencing, for example, okay? So some elements can be difficult to assemble using traditional bulk sequencing, but here, going at the single cell can be easier in certain cases. Capturing genomic heterogeneity, as I mentioned earlier. Studying microbial communities, which is beneficial for when we are sequencing these microbial communities, we can have a better understanding of microbial diversity. This would relate to certain applications, as I will show in one of the coming slides, in terms of antimicrobial resistance, for example, and food, and so on and so forth, energy, all of these interesting discussions. Also understanding genomic mosaicism. Our body might have cells with different genomic content. There are certain diseases or conditions, rare cases, where there might be some different mutated cells in the body. So this kind of single-cell whole genomic sequences can reveal those information. In terms of copy number variations, we know that copy number variations are these segments of the genome that are duplicated or deleted. So some individuals will have different number of copies of that specific genomic region, especially when we have a relative comparison to the parents or other individuals. So this can be detected in individual cells, revealing cell-to-cell genomic heterogeneity, for example. Especially in cancer studies, copy number variations can drive tumor evolution and impact treatment response. Single nucleotide variations, it refers to a change at a single base pair in the genome, only one single base pair. And so detecting these at the single cell as well can reveal somatic mutations as well. So these are going to highlight some of the different applications of whole genome sequencing in pharmacogenetics, okay? And you see here, there's some pipeline in this study for working with the patients, whole genome sequencing, alignment, filtering, interpretation, and then diagnosing and genetic counseling, trying to have a report for each medical safety card. Eventually, we have this documented information about the genes. It's going to be really interesting to have this in the future for every person. Okay, this can you know, everyone is hoping, including me, that we reach this level where we are able to cure diseases like cancer, for example. So having this for every patient, maybe from this card, from early days, maybe from birth, can help in knowing what kind of precautions we may need to go through, some diets, maybe start early screening, and so on and so forth. They will never be conclusive in the sense that sometimes we don't have any mutations and still we get diseases like cancer, but maybe would help in many of the cases and early treatment and so on and so forth. Maybe with this advancements of whole genome, of whole single cell whole genome sequencing, who knows if we end up going to this what's called nano level technologies, nanotechnologies, try to pinpoint some of these cells at early levels. Big struggle we have with cancer when it metastasizes and leaves its position to other organs and so on and so forth. Whole genome sequencing in food industry, as I mentioned earlier, for example, in this case, whole genome sequencing of bacteria has proven useful for tracking diseases, outbreaks, and infection control. There is also what's called whole metagenomic sequencing to study microbial communities and provide data on antimicrobial resistance. And these techniques help in tracing antimicrobial resistant specific genes. Okay, so this can help in, you know, risk assessment and knowing the transmission pathways and energy pathways in food chain. So we went from you know, the overall discussion on genome assembly, clone by clone genome assembly, de novo genome assembly. We discussed the workflows and some in depth, the different steps, the different concepts, how to measure the quality, and so on and so forth. We then went to more focused area, which is, you know, drawing on that genome assembly. What about having now tailored technologies for tailored or specific use cases or applications, which was single cell whole genome sequencing? What about using the same approach and look at a different zoom in strategy, which is not the single cell, but the exome? So this is the whole exome, what's called the whole exome sequencing. And this involves sequencing only the exons. We know that exons, as part of the transcription process, are going to relate eventually to the protein coding regions. or to the protein generation. Exons constitute about what? 1 to 2% of the genome that encodes the proteins. However, something to note, although this is only up from 1 to 2% of the human genome, but it contains approximately 85% of known disease-related variants. So this is a small percentage, but it's very important one for us. So focusing and zooming on this part is like zooming into the diseasing and on the disease and trying to look into disease-causing genes, understanding as well the genetic causes of diseases. So whole exome sequencing uses probes to selectively capture and enrich the exonic regions, and again, trying to achieve deeper coverage, that is more reads per base. It is very focused on this. And again, in terms of library preparation, thinking again about single cell sequencing, it's going to have a specific protocol, specific library preparation. Like in this case, of course, there's going to be fragmentation of genomic DNA. So DNA is sheared into smaller fragments, typically around 150 to 300 to 200 to 300 bases in length, and then trying to repair or apply end repair to have blunt-ended fragments. And after fragmentation, the DNA ends might be overhanging. So the goal of the blunt-ended fragments is to produce fragments with flat or blunt ends that are ideal for what? I'm gonna certainly say, well, can I say here, yeah, I cannot give a treasure hunt question, but the need of this blunt-ended and so forth to make it easier for adapters, okay, to makes it easier for adapter ligation. And there are more steps into this, like addition of A bases to the 3' ends. This is preparing for ligation, DATPs, clinophragments, which is an enzyme used to add an adenine residue using DATP as the substrate, and so on and so forth. So anyway, this would go through this process of ligation and so on and so forth. Eventually, there is this specific protocol or unique way to prepare the library for the whole exome sequencing. And again, we are trying to achieve high coverage, high number of reads, focused on these regions of the exome, as compared to whole genome sequencing. So whole exome sequencing is targeting only again 1 to 2% of the genome, instead of the whole genome sequencing, which is covering the entire genome. Another thing is that whole exome sequencing provides a deeper coverage of these exonic regions, and which offers a comprehensive view of the entire genome. Pardon me. Which while whole genome sequencing would offer a comprehensive overview of the entire genome. So again, whole exome is, we're trying to zoom in into these exonic regions, and we are trying to find the variants in these coding regions, because we know that they're mainly directly related to around 85% or more of the diseases that we know. In terms of data analysis framework, so the whole exome sequencing is going to follow in general very similar sets of steps, and you will see very common tools that we went through before. There might be some differences, but in general, there is the quality control pre-processing as compared to whole genome sequencing, very similar alignment, post-alignment processing, and of course, variant coding can go through different steps and towards annotation. And if you go through this, you will find many of the tools that we already discussed or considered before. This is a visualization of exome sequence alignment, and this is using integrative genomic viewer. We referred to it before, I guess, in the module on databases. So this is a high performance visualization for interactive exploration for the genomic data set. So remember, or let me point out that colors would have specific interpretations here. Red typically represent a paired end read, where the mate pair, the other end of the fragment, is mapped to a position that is left of the current view, or is mapped to a different chromosome. In other words, this can indicate an inversion in some cases. And again, these colors depends because you can even introduce changes in different interpretations. A green, on the other hand, typically represent the paired end read, where the mate pair is mapped to a position that is to the right of the current view. Okay. Okay, and then yes, so we are trying to see here the mapping of these aligned regions and get focus on these exons and try to see if we have any differences, any inversions, any deletions, and so on and so forth. This summarizes the differences when we look at structural variant, comparing whole genome sequencing to whole exome sequencing. Whole genome sequencing compared to the whole exome sequencing. So this is trying to cover all the variants across all the reads. So we have more comprehensive coverage, but when it comes to whole exome sequencing, we are focused on specific regions. So I'm going to stop here and we'll continue in the next part.